In biology, a species complex is a group of closely related species that are very similar in appearance to the point that the boundaries between them are often unclear. Terms sometimes used synonymously but with more precise meanings are cryptic species for two or more species hidden under one species name, sibling species for two cryptic species that are each other's closest relative, and species flock for a group of closely related species living in the same habitat. As informal taxonomic ranks, species group, species aggregate, and superspecies are also in use. Two or more taxa once considered conspecific may later be subdivided into infraspecific taxa, but this is not a species complex. A species complex is in most cases a monophyletic group with a common ancestor, although there are exceptions. Hybrid speciation can be a component in the evolution of a species complex. Species complexes exist in all groups of organisms. They are identified by the rigorous study of differences between individual species, making use of minute morphological details. Tests of reproductive isolation, or DNA-based methods such as molecular phylogenetics or DNA barcoding. The existence of extremely similar species may cause local and global species diversity to be underestimated. Recognizing similar but distinct species is important for disease and pest control, and in conservation biology. Although drawing dividing lines between species can be inherently difficult. Definition: a species complex is typically considered as a group of close, but distinct species. Obviously, the concept is closely tied to the definition of a species. Modern biology understands a species as separately evolving metapopulation lineage, but acknowledges that the criteria to delimit species may depend on the group studied. Thus, many species defined traditionally, based only on morphological similarity, have been found to comprise several distinct species when other criteria, such as genetic differentiation or reproductive isolation, were applied. A more restricted use applies the term to closed species between which hybridization occurred or is occurring, leading to intermediate forms and blurred species boundaries. Some authors apply the term also to a species with intraspecific variability, which might be a sign of ongoing or incipient speciation. Examples are ring species or species with subspecies, where it is often unclear if these should be considered separate species. Included concepts: several terms are used synonymously for a species complex, but some of them may also have slightly different, or more narrow meanings. In the nomenclature codes of zoology and bacteriology, no taxonomic ranks are defined at the level between subgenera and species, while the botanical code defines 4 ranks below genera. Different informal, taxonomic solutions have been used to indicate a species complex. Cryptic species, also physiologic race. This describes distinct species that are erroneously classified under one species name. More generally, the term is often applied when species, even if known to be distinct, cannot be reliably distinguished based on their morphology. The usage physiologic race is not to be confused with physiological race. Sibling species, also aphanic species. This term, introduced by Ernst Mayr in 1942, was initially used with the same meaning as cryptic species, but later authors emphasized the common phylogenetic origin. A recent article defines sibling species as cryptic sister species, meaning two species that are the closest relative of each other and have not been distinguished from one another taxonomically. Species flock, also species swarm. This refers to a monophyletic group of closely related species all living in the same ecosystem. Conversely, the term has also been applied very broadly to a group of closely related species that can be variable and widespread. Superspecies, sometimes used as an informal rank for a species complex around one representative species. popularized by Bernhard Rensch and later Ernst Mayr, with the initial requirement that species forming a superspecies must have allopatric distributions. For the component species of a superspecies, allospecies was proposed. Species aggregate used for a species complex, especially in plant taxa where polyploidy and apomixis are common. Historical synonyms a species collective are, introduced by Adolf Engler, conspecies, and Grex. Components of a species aggregate have been called segregates or microspecies, used as abbreviation AGG, after the binomial species name, sensu lato meaning, in the broad sense. This Latin phrase is often used after a binomial species name, often abbreviated as SL, to indicate a species complex represented by that species. Identification. Distinguishing close species within a complex requires the study of often very small differences. Morphological differences may be minute and only visible using adapted methods, such as microscopy. However, distinct species may sometimes have no morphological differences. In these cases, other characters, e.g., in the species' life history, behavior, physiology, or karyology can be explored. As an example, territorial songs are indicative of species in the tree creepers, a bird genus with little morphological differences. Mating tests are common in some groups such as fungi to confirm the reproductive isolation of two species. Analysis of DNA sequences is becoming increasingly standard for species recognition and may in many cases be the only useful method. Different methods are used to analyze such genetic data, for example molecular phylogenetics or DNA barcoding. Such methods have greatly contributed to the discovery of cryptic species, including such emblematic species as the fly agaric or the African elephants. Evolution and ecology. Speciation process. Species forming a complex have typically diverged very recently from each other, allowing in some cases to retrace the process of speciation. Species with differentiated populations such as ring species are sometimes seen as an example of early, ongoing speciation, i.e., a species complex in formation. Nevertheless, similar but distinct species have sometimes been isolated for a long time without evolving differences, a phenomenon called morphological stasis. As and examples, the Amazonian frog, Pristimantis ocendini, is actually at least 3 different species that diverged over 5 million years ago. Stabilizing selection has been invoked as a force maintaining similarity in species complexes, especially when adaptation to special environments, such as a host in the case of symbionts, or extreme environments, constrains possible directions of evolution. In such cases, strongly divergent selection is not to be expected. Also, asexual reproduction, such as through apomixis implants, may separate lineages without producing a great degree of morphological differentiation. A species complex is usually a group that has one common ancestor, although closer examination can sometimes disprove this. As an example, the yellow-spotted fire salamanders, in the genus Salamandra, formerly all classified as one species S. Salamandra, are not monophyletic. The Corsican fire salamander's closest relative was shown to be the entirely black alpine salamander. In such cases, similarity has arisen from convergent evolution. Hybrid speciation can lead to unclear species boundaries through a process of reticulate evolution, where species have 2 parent species as their most recent common ancestors. In such cases, the hybrid species may have intermediate characters, as demonstrated e.g. in heliconius butterflies. Hybrid speciation has been observed in various species complexes, such as insects, fungi, and plants. In plants, hybridization often takes place through polyploidization, and hybrid plant species are called nodar species. Range in regards to whether or not members of a species group share a range. Sources differ. A source from Iowa State University Department of Agronomy says that members of a species group usually have partially overlapping ranges but do not interbreed with each other. A dictionary of zoology describes a species group as complex of related species that exist allopatrically and explains that this grouping can often be supported by experimental crosses in which only certain pairs of species will produce hybrids. The examples given below may support both uses of the term species group. Often such complexes only become evident when a new species is introduced into the system, breaking down existing species barriers. An example is the introduction of the Spanish slug in northern Europe, where interbreeding with the local black slug and red slug, traditionally considered clearly separate species that did not interbreed, shows they may be actually just subspecies of the same species, where closely related species coexist in sympathy. It is often a particular challenge to understand how these similar species persist without outcompeting each other. Niche partitioning is one mechanism invoked to explain this. Studies in some species complexes indeed suggest that species divergence went in par with ecological differentiation, with species now preferring different microhabitats. Similar methods also found that the Amazonian frog Eleutherodactylus ockendenai is actually at least 3 different species that diverged over 5 million years ago. A species flock may arise when a species penetrates a new geographical area and diversifies to occupy a variety of ecological niches. This process is known as adaptive radiation. The first species flock to be recognized as such was the 13 species of Darwin's finches on the Galapagos Islands described by Charles Darwin. Practical implications. Biodiversity estimates. It has been suggested that cryptic species complexes are very common in the marine environment. Although this suggestion came before the detailed analysis of many systems using DNA sequence data, it has been proven correct. The increased use of DNA sequence in the investigation of organismal diversity has led to the discovery of a great many cryptic species complexes in all habitats. In the marine bryozoan cell Phora hyalina, detailed morphological analyses and mating compatibility tests between the isolates identified by DNA sequence analysis were used to confirm that these groups consisted of more than 10 ecologically distinct species that had been diverging for many million years. Evidence from the identification of cryptic species has led some to conclude that current estimates of global species richness are too low. Disease and pathogen control. Pests, species causing diseases, and their vectors, have direct importance for humans when they are found to be cryptic species complexes. The ecology and virulence of each of these species needs to be re-evaluated to devise appropriate control strategies. An example are cryptic species in the malaria vector Anopheles, or the fungi causing cryptococcus. Conservation biology. When a species is found to comprise in fact several phylogenetically distinct species, each of these typically have smaller distribution ranges and population sizes than reckoned before. These different species can also differ in their ecology, e.g., having different breeding strategies or habitat requirements, which has to be taken into account for appropriate management. For example, giraffe populations and subspecies differ genetically to such an extent that they may be considered species, while the giraffe as a whole is not considered threatened. Considering each cryptic species separately would mean a much higher level of threat.